This is a situation where there's what we refer to as kind of strict dominance recessiveness. Where in this case, this allele masks the action of this allele. So the only way that this allele can show itself, can express itself, is when it's not in the presence of the other allele. So I gave you this definition before, but a dominant trait, a dominant allele, expresses itself whenever present. And a recessive allele is only expressed in the absence of the dominant allele. So the reason that I say it this way, I used to say that a recessive trait was only expressed when it's in the homozygous condition. But remember that in human beings, we have sex chromosomes. And the sex chromosomes usually have an X chromosome and what we refer to as a Y chromosome. If you're a male, your sex chromosomes are this. In humans, it's different in birds, it's different in amphibians and reptiles. But in humans, the male has an X chromosome and a Y chromosome, and this Y chromosome is reduced in size. And we saw this back when we looked at the chromosomes earlier in the semester. And the reason that this Y chromosome looks this way is because it doesn't have all of the genes on it that are on the homolog. So this is a pair of homologous chromosomes, just like we have for all the other chromosomes. But in this case, this chromosome is missing like half of what would normally be on the X chromosome. And so in this case, we have genes that occur on this half that's essentially kind of missing from the Y chromosome. And so even though there may only be one allele here, because there's not a dominant allele on the other chromosome, because there is no corresponding part of that chromosome, this is the thing that gets expressed, because it isn't that it's homozygous, it's just that it's not there. This is a condition called hemizygous, half-zygous. And so since that's the only allele that's there, it ends up getting expressed. This is why we have certain genes that appear, for example, only in males. Because if an allele appeared in a female, you would be likely to get a dominant allele on that other chromosome. That would then show the dominant trait. And so certain traits appear more frequently in males than in females, because you get the recessive allele from your mother, you don't have a dominant allele to counteract it because you don't even have that section of the chromosome, so then you get the recessive trait being expressed, yeah.
So is that like being colorblind? Because don't you?
That is exactly like being colorblind. It's also being hemophilia. One of the types of hemophilia is an excellent trait, and males tend to be affected more by hemophilia than females. And it's the same sort of situation. So another way of saying a recessive allele is an allele that expresses itself only in the homozygous recessive state or in a hemizygous state. But then a simpler way of just saying that is it's just in the absence of a dominant allele that the recessive allele gets expressed. So in strict dominant recessive traits, this is how things work. But we also have what's known as incomplete dominance. This is something worth noting about plants because some plant species have different colors of flowers. So I believe this occurs in a plant called adipinuts. This is a flower that either has a red flower, a pink flower, or a white flower. The red flower has 2 copies of the dominant allele. They're homozygous dominant. The pink flower is heterozygous and the white flower is recessive. And the reason that this comes about is that this is essentially a dosage effect. The recessive allele produces no red pigment. The dominant allele produces essentially like one dose of red pigment. And this guy produces 2 doses of red pigment. And so the more red pigment that you produce, the redder you are. And so if you produce 1 dose of red pigment, you end up being a pink flower. If you produce 2 doses of red pigment, you end up being a red flower. But the thing about this is that's weird compared to, for example, round versus wrinkled peas, is that you take a pure breeding red plant, cross it with a pure breeding white plant. This is a red plant, this is a white flowering plant. What kinds of gametes can this individual produce? Just the dominant allele. What type can this produce?
Recessive.
Just the recessive type. When we bring those together, we get a heterozygous pink plant. Neither of those phenotypes appeared in the parents. When we then cross these pink parents, allow these pink offspring rather to cross somebody, Dakota, come up here and do this cross. Okay, Mr. Espinoza, give us the genotypic and phenotypic ratio.
It's going to be.
Go ahead and go right on the board. Hmm? There's 1 genotypic ratio and there's 1 phenotypic ratio. So the genotypic ratio is just the genotypes and what frequency they occur in. So you have how many genotypes? Yep.
So what's that going to be
2 over 4, sure. 2/4 of them are what?
Dominant.
Not
Or
dominant.
dominant recessive.
Which is a heterozygote, yep.
Okay.
So the genotype is big R, little r.
Okay.
1/4 is
recessive.
little r, and how many quarters is the homozygous dominant?
1.
Yeah. So since the denominator is all the same, you can just say for every 1 homozygous dominant, we have 2 heterozygotes and 1 homozygous recessive. We typically write it 1, 2 to 1, with these being them. If you put that on an exam, I would grade that correct because those are the correct ratios of those 3 genotypes. You didn't do phenotypes though, so call on somebody to do the phenotypic ratio. Who? Okay. So this was the genotypic ratio. Phenotypic ratio. Awesome, you saved yourself some time because the genotypic ratio and the phenotypic ratio in incomplete dominance is the same. You have 1 red flower to 2 pink flowers to 1 white flower. So this is a situation where the more copies of the dominant allele you have, the stronger the phenotype is. The more of the phenotype is that you get. And so 2 dominant alleles, a lot of the phenotype. 1 dominant allele, less of the phenotype. Homozygous recessive, none of whatever the phenotype is. So these are just white flowers that lack any sort of pigment whatsoever. They just don't have a pigment, so they don't produce any sort of color as a result. gene action or inheritance that we want to talk about is codominance and multiple alleles. This has less to do with plants and more to do with humans, so I want to mention it. So far, we've been looking at single gene, 2 allele systems, a dominant allele and a recessive allele. But there are traits that have multiple alleles, lots of different alleles. This is the case in humans with blood type. Do you guys know what your blood type is? No, you should know your blood type so if you get in an accident, you can tell people your blood type.
Does the hospital know your blood type?
Why would the hospital know your blood type?
I haven't gone to the hospital.
Mine's a
I haven't
positive.
gone to the hospital.
Yeah, like, so I bike, you guys know that I bike. I was in Wichita Falls in Texas in 2013, competing in a thing called the Harder Than Hell 100, and I crashed in mile 7 of a 100-mile race and broke my collarbone, banged up my hip, was knocked out on the side of the road, ambulance came, et cetera, et cetera. This is a thing called road ID that I wear pretty much constantly. It basically has emergency contact information on it, but also first responders can basically phone the number on the front and then give them the serial number and the PIN number that's on the back of this. It has all my health information on it, including prior injuries, things like blood type, et cetera, et cetera. Am I asthmatic? I am not. Am I allergic to any drugs? I am not. So that when I'm unconscious on the side of the road, they don't have to ask me what my blood type is. If I need a transfusion there, they can just find out from a company. So it's good to know what blood type you are, but you guys don't know for the most part?
Where did you get that?
Oh, it's from a company. A bicyclist guy was injured, and then he was unconscious, and they couldn't get any of his health information because he was unconscious. So we came up with the idea of creating a way that athletes could store their information online securely, and these little bracelets just link you to your account. And so it's good to have. I'm not advocating that anybody do it. It's just for me as a bicyclist, it's a little peace of mind. Also, it has emergency contact information right on there, so that when something does happen to me, they have the phone number of the person that I want them to call first on it. Blood type. Do you guys? So you don't know what your own blood types are. What are the variety of blood types you could have?
Ain't it old or something?
So O is one of them.
A,
AB. We won't worry about positives and negatives, that's actually a different gene. So these things refer to blood type. And if you look at your cell, there are little complexes of proteins and sugars that adhere to the outside of your cells. And these things are called antigens on a red blood cell. And these antigens are involved in your body's recognition of self. So you have immune cells that recognize foreign substances coming into your body, foreign things like bacteria and things like that coming into your body, and they will react and mount immune responses to those foreign things. But the way they know that those foreign things are foreign is because they have antigens that are different from your antigens. And so these antigens are used in your body's immune system's way of recognizing that your cells are your cells. When you have an autoimmune disease, your immune system basically refuses to acknowledge that your cells are your own and attacks them as if they were an invading pathogen, and then it kills cells that you don't want to kill off. So in this case, you can have red blood cells that have A antigens. Type B blood has B antigens. AB blood has both A and B antigens. And O blood has no antigens.
Ain't O the rare one or something?
I don't remember what the frequency of them are. We could look that up. I just don't remember. So these antigens on your blood cells are coded for by genes. And so this is hereditary. You inherit your blood type from your parents' blood type. Well, as it turns out, the A antigen is coded for by a dominant allele. The B antigen is coded for by the dominant allele. Having no antigens is just like having no pigment. The allele just doesn't produce any antigens. And so if you are type O blood, we know that you're homozygous recessive because you're not producing any of these antigens. So if you have A antigen, you can either be a homozygous dominant or you can be a heterozygous. You don't have to say heterozygous dominant. Heterozygous implies that there's a dominant allele there. B antigens would be homozygous dominant or heterozygous. Yeah, there you go. What would somebody with AB blood be genetically? Genotypically? Somebody said something. If you have both A antigens and B antigens, it would be heterozygous. This way. And in this case, this is heterozygous dominant, if you wanted to say it that way. I would just say it's heterozygous, but it's heterozygous in a way that is different from being this heterozygote. This is a heterozygote with codominant alleles. Codominance means that you have two dominant alleles, both of them get expressed if they're present. And so this is where blood type comes from, and it leads to interesting patterns of inheritance because if you have a type A mom and a type B dad, what are the possible genotypes that these two individuals could be? They could be this, or they could be that. This individual could be this, or it could be that. What do we do when we don't know? Nope. We put a little space there because we know that a type A person has to have at least one dominant A allele. Type B person has to have at least one dominant type B allele. When we go to cross these, if we look at these people's offspring, there are two outcomes, aren't there? If these guys, well actually there are more than two outcomes. If these guys produce nothing but AB offspring, there's only one genotype here, right? And let's say that they have 5 offspring, and every one of them is type AB. What would you infer about the genotypes of the parents? You would assume that they're both homozygous because after 5 children, the chances of having that many offspring that are all AB becomes kind of small. Because if these guys were heterozygous, what would you expect? Sydney, come do the cross. I knew it was coming eventually. This is how you get good at crosses, by practicing them. Which is why I'm doing this to you guys. Is that for a B or an A?
This is A, this is B.
What do you have? Oh, is that an A? It's hard to see.
Yeah, sorry. Yeah.
Okay. Okay, pick somebody to come up and give us the genotypic and phenotypic ratios. Somebody who hasn't gone yet.
Alright, you wanna go? Sure.
Do genotypic ratio and then call on somebody with your phenotypic ratio. So we'll force you guys to know one another's names also. Do you guys know one another's names? Oh, no. it's very important. What is she doing that you should always do with genotypic and phenotypic ratios? So you're not only giving us the numerical ratio, but you're also referring that ratio to what that ratio refers to. In this case, the different genotypes. So you have a 1:1:1:1 ratio of heterozygous with both dominants, heterozygous with dominant B, heterozygous with dominant A, and homozygous recessive. Who do you want to give us the phenotypic ratio?
I'm lost. There's no way. I'm
Okay.
sorry. I don't know. I don't know.
Go
I have no idea. I literally have no idea. I'm lost.
Guess what? You
What?
have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 people in the room to help you. Kaden, to the board. If you have a dominant A and a dominant B allele, what?
Look at the board, dude.
Look at the board.
Okay.
What
Look
is-
at the board, dude.
Okay.
I didn't say it first. Somebody out there said it. If you have that first genotype, what's the only phenotype you can have?
I'm still lost. Yeah, I mean, like, type AB would.
Yes, it's type AB blood. So,
Well, I mean, like, how do I literally have no idea. I'm really just lost.
so these, she didn't put colons in here
Okay.
separating them. So
So
this is a
the-
genotype, that's a genotype, that's a genotype, that's a genotype. So you have 1/4 of the population should be, 1/4 of the offspring should be type AB.
And then.
1/4 should be, what's the next genotype?
B.
Type B.
Yep. And.
What's the next genotype?
A.
And what's the next genotype?
O.
That is O.
I don't know, I just write down the letters, dude, I don't know.
No antigens, no antigens is type O. A antigens only is type A, B antigens only is type B. Both types of antigens maybe. You did it. So if you have 5 children and they're all type AB, that tells you that you have a lot of homozygosity in the parents. You're only going to be getting lots of different phenotypes out of offspring if there's some degree of heterozygosity in the population. I could just as easily have given you one of these guys is homozygous dominant for blood type and the other is heterozygous. And what would you do? How would this change?
You would get more A's.
You would lose this right here, right? Because you wouldn't have that recessive allele coming from this individual. This individual can only produce this allele, so you would only expect 2 phenotypes in your offspring, type AB and type A. You wouldn't expect type B blood, and you wouldn't expect type O blood out of that cross. So those things lead to kind of what we think of as non-Mendelian sorts of ratios of genotypes and phenotypes. It's not a big deal. You use the same mechanics to work the cross. Figure out what the gametes are, throw those gametes into a Punnett square, do all the combinations, and figure out what the genotypes and phenotypes are. Same process, but in this case we have multiple alleles and we have multiple phenotypes as a result of that. Same process. Questions? Okay. If you can do this with single genes, whether they have 2 alleles or multiple alleles, you can do this with 2 genes. Or 3 genes, or 4 genes, or 5 genes, or 10 genes, or 20 genes. So, let's take up the case of brown seeds that are yellow crossed with wrinkled seeds that are green. And these guys are going to be pure breeding, and so that means that they're homozygous for both of these traits. So pure breeding would be this, and pure breeding would be this. Now, when you go to make gametes for these guys, how do we make gametes? Gametes are haploid, so remember these guys are diploid. Why are they diploid? because we have how many copies of each gene? We have 2 copies of each gene. So the gametes are going to be haploid, which means that they have 1 copy of each gene. Because the alleles are the same here, this individual can only produce 1 allele of the round gene, and can only produce 1 allele of the yellow gene. So their gametes will look like this. There are 2 traits in the gametes now, rather than 1 trait before, because now we're dealing with 2 traits. This individual can only produce 1 type of gamete for each of those things. When fertilization occurs, what's the genotype of the new offspring?
Big R, big Y, little r, little y.
Heterozygous for the first trait, heterozygous for the second trait. Phenotype,
Round, yellow,
the dominant trait is the trait that comes out in the F1 offspring. Round yellow. But now they're heterozygous. If we then allow these pea plants to self, life starts to get a little more complicated. How do we go about finding gametes for a dihybrid cross? A cross with 2 traits.
Double Punnett square.
It's a Punnett square, but it gets bigger. But the first thing we have to do, what's the first step always? Figure out what the gametes are. How would we determine gametes? Yeah.
Is it still gonna be the 3/4 dominant, 1/4?
Those are ratios. Step back to gametes. If we have this genotype, how do we figure out what the gametes are going to be that this individual can produce? This individual has to make gametes. What will they be?
It's gonna be
Big R, big Y.
Big R, big Y? Another gamete type?
big R, little y.
Big R, little y?
Little R, big Y.
Little r, little y.
Okay, how'd you do that?
Foil.
You did foil. Kate, what's foil?
Like-
Come up here and explain it.
Jesus, alright.
You know something. so
Yeah,
don't
no, I-
find out when you know something. You should be
I was
happy
gonna
about
point the
that.
first part.
Explain it to us.
Okay, so what I do is I start with like the big first letter, and then I kind of multiply it first to here, and then first to here. So first one, big R, big Y.
So you called it foil. Why did you call it foil? What does foil stand for?
Yeah.
Dude, I don't know. I just know it's this the process you do it. So first,
First.
yeah.
No,
Okay, there you go.
Right?
Yes.
Foil, first outer inner last. Where did you learn foil? Math. If you want to take a plus b and multiply it times x plus y, what do? Multiply first, outer, inner, last. Because it does all combinations of the multiplication across this polynomial. Same thing here. Do the first, do the outer, he didn't finish, do the inner, and then do the last. And you'll get those things in that order every time. Caden says he doesn't know anything, he knows stuff.
No, I was little like scattered the
Yeah.
first part of class. So I wasn't
This
paying
is how you
attention.
make gametes in dihybrid crosses. It's the easiest way to go about doing it. Just apply the FOIL method. Well, if this individual can make those gametes, this individual can make those gametes. And so this gives us a Punnett square that now is more complicated, but it's the same process. We have to be a little more careful as we track things across the Punnett square, but the Punnett square is also kind of magical in this way. I will show you the magic of the Punnett square. Whoop, I need to draw. Always try and draw your dominant and recessive alleles noticeably different. What do we do when we work a Punnett square? We combine gametes. So to keep things straight, I try and do things systematically. So I'll take these guys and I'll bring them across. And you see that I'm leaving space for other alleles to come in. So I've brought all of these alleles across. Each row looks the same all the way across. Once you've brought all those alleles across, you can bring these alleles down and fill in. Easy peasy. It's the same thing as a 2x2 when you're dealing with one trait. When you're doing a 4x4 with two traits, you just bring some alleles across, you bring the other alleles down. But now things get more complicated because who wants to do the genotypic ratio for this cross?
Charlie.
Somebody who hasn't gone yet.
Charlie's got it.
Charlie, you want to do it?
He wants to do it.
Start at the upper left-hand corner here. You have a genotype, right? Homozygous dominant for both traits. Right?
Yeah.
How many of this genotype are in that table?
One.
Then, what do you notice right here on this diagonal? They're exactly alike. Sounds like Darren wants to do this. So we have two of those, right? Then we have another diagonal here. What do we find in that diagonal?
Three of them.
They're not three alike.
Just two.
There's one of them that's different and two of them that are alike, right? There. Continue with this diagonal. Come on. Everybody's going to the board by the end of the day today.
Gone twice already, man.
Some people have went twice, maybe 3 times, Caden.
Shit.
What do you have now, this diagonal?
2 and 2.
2 and 2?
These two. Yeah, those two.
Are they all the same? Or are they different?
These two are alike. These two are too. So it's 2 and 2, right?
Are they all the same all the way across the diagonal?
This one right here.
Yeah, they are all the same or are they different?
Are they all the same?
They're all the same, so you have 4 of those. They're heterozygous for both traits. Nope. Heterozygous for both traits. You have a recessive allele for the second gene. So heterozygous and heterozygous. So little y for the second one.
So like big R, little R.
Dominant and recessive for the first trait and dominant and recessive for the second trait. And there are 4 of those.
Last one's a lot.
Oh yeah, the last one is y. Always keep your genes together. So in this diagonal, what do you have?
I got 2 and 1.
You've got 2 that are this genotype, so put those up there. Homozygous recessive for the second trait. Yep, homozygous recessive. That's a little y, little y.
For this.
Yep, little y, little y, little y, little y.
Alright.
Notice this is similar to that, except flipped. You have homozygous dominant for the first trait, homozygous recessive for the first trait,
What do we do when it's like that?
homozygous recessive for the first trait, homozygous dominant for the, so there's just one of these, but it's homozygous recessive for the first trait and homozygous dominant for the second. Okay, what do you have for this diagonal? Homozygous recessive for the first trait and heterozygous for the second trait, and there's 2 of those.
Yeah.
And then this last one is homozygous recessive for both traits. What is Punnett square?
Tells you everything.
Well, it tells you everything, of course. It tells you everything about this particular cross vector. So these are the genotypes. So this is the genotypic ratio. What do you notice about the genotypic ratio? 1 to 2 to 1, to 2 to 4 to 2 to 1 to 2 to 1. It's symmetrical, isn't it? This is the middle part. This is the middle of the Punnett square. It's the same above and to the left as it is below and to the right. The only thing that changes is the genotypes. You notice that there's symmetry and that this is similar to that, only flipped. This is just like that, except that in this case it's homozygous dominance for both traits and homozygous recessives for both traits. There is a symmetry to the Punnett square when you do your gamete formation using the FOIL method and list them in order, you get this symmetry that comes out of it. This symmetry is true for 2 traits. It's also true for 3 traits. So now we have all of these genotypes. And the genotypic ratio is a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio for these genotypes. Aaron, pick somebody to come and do the phenotypic ratio. How many phenotypes do we have? Not the same amount of genotypes.
That's a 9 right there, but there's 4.
So we can actually figure this out also, because you can either be round or wrinkled for the first gene, or you can be yellow or green for the second gene. How can we figure out how many genotypes we have? Dayton? We have a binomial here, right?
Okay.
Somebody over here said foil.
Well, yeah.
We can have round yellow.
Dude, I'm so lost.
We can have round green. We can have wrinkled yellow. Or we can have wrinkled green. These are all the possibilities. So each of these genotypes have to map into one of these phenotypes, right? So what's the phenotype of this genotype? Brown yellow. How many do we have?
1.
What is the phenotype of this genotype?
Brown
And we have 2 of those. What is the phenotype of this genotype?
Brown, green.
And there's 1 of those. What's the phenotype here?
Brown, yellow.
Phenotype here?
Brown, yellow.
Phenotype here?
Brown, green.
Green.
Phenotype here?
Wrinkled yellow.
Wrinkled yellow.
Phenotype
Wrinkled green.
here?
Wrinkled yellow.
Thank you. Look at that kid.
Yeah. I know. Huge.
Well, this is plus,
Yeah, no,
plus-
wait,
plus-
wasn't the
plus 2.
R yellow, wasn't that wrinkled green above it? Yeah,
This is
what?
wrinkled green.
Yeah, you don't have it labeled. Wrinkled green.
No, this is wrinkled yellow. Wrinkled yellow.
Wait, what?
Those are
It-
two-
Those are
those
capitalized.
no,
Those
oh,
are
okay,
capitals.
okay. They look yellow.
Those are
Okay.
capitals. That's why I always do a curly Q for my lowercase, because it helps me.
I can't see that. I'm just blind.
Yeah.
My handwriting ain't the best.
Yeah, bro, I don't even.
So-
You're good, dude. You're good.
So the only genotype we have left is this one right here, right? Which this is
Wrinkled green.
wrinkled green. 9, 3, 3, 1. This is the ratio of round yellow to round green to wrinkled yellow to wrinkled green. Most of them, 9 out of 16, slightly over half of them, are the dominant phenotype because the dominant allele is the one that gets expressed when it's present. The wrinkled green only occurs when both alleles are recessive for both genes. So there's only 1/16 of the population. So this thing should add up to 16, 9, 10, 11, 12, 13, 14, 15, 16. That should add up to 16. So this is also the same thing as saying 9/16 of the population is this, 3/16 of the population is that, 3/16 and 1/16 is that. So the phenotypic ratio is this. And so if you planted these plants as pure breeding plants, got the F1s and then allowed them to interbreed with one another, this is the number of peas that you would expect in your population in peas to have these two traits. And it doesn't matter whether it's peas that you're talking about, or humans, or cows, or wheat, or whatever, all things would adhere to these rules. But once again, working it out in the two-trait case is not any different than working it out in the one-trait case. It's just that you have to now form gametes, each gamete having two genes, two alleles in each gamete. And so the method of figuring out the gametes is a little more complicated. The Punnett square gets bigger, but it's the same process. If you can do a one-trait Punnett square, you can do a two-trait Punnett square. It's just that a one-trait Punnett square is 4 blocks big. This one is 16 blocks big. Do you feel confident about a two-trait Punnett square? So let's see if we got it right. This is basically from your textbook, this section on genetics. There's our Punnett square. There are all of our things. Round yellow, 1, 2, 3, 4, 5, 6, 7, wait, 1, 2, 3, 4, 5, 6, 7, 8, 9. Wrinkled yellow, 1, 2, 3. Round green, 1, 2, 3. Wrinkled green, 1. Genotypic ratios and phenotypic ratios, well, it just gives you the phenotypic ratio there. When Mendel did this, he basically performed this exact trait back in the 1850s. And what he found was that when he got the F2 generation, this is the F2 generation. We had the parental generation, then we had the F1. We cross the F1s, allow them to self-pollinate, and we'll get the F2. 556 seeds total, 315 of them were round yellow, 101 were wrinkled yellow, 108 were round green, and 32 were wrinkled green. 9.8 to 3.16 to 3.36 to 1. Those actually are statistically not different from a 9:3:3:1 ratio. And so it works on a board. It also works when you're actually planting seeds out in the world. Questions?
Well, would that account for more than what we got? Like 9.8 plus.
Yeah, so there's going to be variability because these are actual seeds, and not all seeds that are fertilized necessarily get to the seed stage so that you can plant them and figure out their phenotype, things like this. So there's some wiggle room in the data, but if you did a chi-square test on this, which we can do really quick, so we have 556 seeds, 315 were round yellow, 101 were wrinkled yellow, 108 were round and green, and 32 were wrinkled and yellow. You guys know how to do a chi-square test? Ooh, I'm going to show you something. Let's go and get a.
What's it called? What's it called?
A chi-square test is just a test to test whether or not two distributions are similar to one another or not. So what we can do really quickly here is we have 4 phenotypes: round yellow, wrinkled yellow, round green, wrinkled green. The observed was 315, 101, 108, and 32, for a total of what we would expect, based on what we know of Mendelian genetics, would be equals 9/16 times 556. So we would expect to see 312.75 if this were working. We would expect 3/16 times 556, which would be 104.5. We would expect the same thing for this. We would expect 1/16 times 556 for that. And that would hopefully add up to 556 also. So what a chi-square test does, and this is a test that we use in genetics, is it compares the observed and the expected with one another, so it subtracts the expected from the observed. Because in some cases, the expected will be above the observed, in some cases it will be below the observed. We square that, we divide it by the expected value, and we sum this up across all of our categories. And so we can calculate this pretty easily in Excel, because we can just write a little equation, equals the observed minus the expected squared divided by the expected. That's what that is. We can copy that formula down. We can sum that up. And so the chi-square statistic in this case is 0.47, essentially 0.47. We can then use the power of the internet to visit Daniel Soper's website, Daniel Soper chi-square. And we can get a p-value calculator and see if this is a significant p-value. And so we have a chi-square value of 0.47 for 3 degrees of freedom, because we have 4 categories, and we find a serious lack of significance. P is would normally need to be below 0.05 for something to be deemed statistically significant. The P in this case is 0.92. We're not anywhere close to being significantly different from a 9:3:1 ratio. So even though those numbers are different than a 9, a 3, a 3, and a 1, they're not so different that we would say that they're different from what we would expect. Does that make sense? So Mendel basically found, effectively, a 9:3:1 ratio. And the nice thing about genetics is it's pretty quick and easy to just do a quick test on does this ratio differ from a 9:3:1 ratio? We can do the math and figure it out if it does or not. In this case, it doesn't. Other questions? All right. Well, if we can do it with 2 traits, we can do it with 3 traits. So let's do it with 3 traits. Let's say that we're interested in 3 genes, gene A, gene B, and gene C, and we cross 2 pure breeding individuals with one another. What are the gametes that this individual can do? This is the parental generation. What gametes can this individual produce?
A, B, C.
A big A, a big B, a big C. What gametes can this individual produce? Little a, little b, little c. You fertilize those guys. What are the genotypes of all of the offspring going to be? Heterozygous for all 3 traits, right? Everybody with me so far?
Yeah.
Kidding?
Yeah.
Yeah? Okay.
I said yeah.
All right, I couldn't tell if it was yes or no.
Oh, we're good.
So if we then allow those guys to self-fertilize, how figure out the gametes that this individual can make?
Make a big square.
Well, let's find gametes first.
Foil.
Okay, what are we gonna foil?
Maybe big A, little a with the 3 traits on the right, and then you do big B, little b times the 3 traits on the right.
Simpler way of doing it. We already know how to foil this, right?
Yes.
Right?
Yeah.
But we have to do that once for
B, B, C, C.
the big C,
Yeah.
and then we have to do that another time for the little c, right?
Yes.
And these are the gametes for this individual, and we'll have those same gametes for that individual. 1, 2, 3, 4, 5, 6, 7, 8. 8 gamete types times 8 gamete types. Our Punnett square is now 64 squares big. So I want you to take a few minutes and fill out the Punnett square. You have paper in front of you? Oh my God, you guys think I'm serious. I'm not serious.
I was really gonna do it.
I've already done it for you because it's a pain in the butt to do this. But what do you notice? So all I did was I brought the gametes across, and then I brought gametes down. What do you notice about this Punnett square? It's symmetrical. Up here, homozygous dominant for all 3 traits. Homozygous recessive for all 3 traits. What do you notice about this diagonal? Same genotypes. What do you notice about this diagonal?
2 to 1.
2 of those, 1 of those. What do you notice about the big diagonal going all the way across the middle? They're all the same. Same rules apply. Doesn't matter if you're doing it 1 trait, doesn't matter if you're doing it 2 traits, doesn't matter if you're doing 3 traits, 4 traits, 5 traits. Making gametes gets more complicated, the Punnett square gets more complicated, but it's the same idea. So, I don't know how they feel about me writing on their screens, but I don't really care. We've been studying traits that are kind of these either or traits. You either have round seeds or you have wrinkled seeds. You either have yellow seed coats or you have green seed coats. Those are one type of trait. You either have A antigens or you have B antigens or you have no antigens. Get up for a minute. Stand along that wall. Put the tallest person at the front of the room, the shortest person at the back of the room.
How tall are you?
I'm 5'8".
The heck is it?
Same. We're gonna be farther back.
This should be easy. You can look at one another and see who's taller.
I feel like we're
Brandon
the
is
same
the
height.
tallest.
I know, that's why I was looking at it. I was like, hey.
Are you taller than me? I think you probably are.
Right next to
I guess.
Yeah. Screw that.
Who's taller? I feel like you taller.
You two, I don't know.
That's what I'm saying. I feel like you taller though.
I don't know.
No, that's how.
We're like all 3 like.
I'm not in the middle.
Yeah, hop in the middle. That'll be close
No,
enough
how
for
tall are
20,
you?
20.
5'11".
Oh, sadness.
Yeah, but I feel like.
Are you always at the end?
Yeah.
What do you notice about height?
Varies.
It varies, doesn't it? We don't just have tall people and then short people. In pea plants, we have tall plants and short plants and nothing in between, because height in pea plants is one of these either or kinds of traits. Height is not an either or kind of trait. We have tall people, then we have slightly shorter, 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 slightly shorter Dakota.
It is so big.
Here's this, sorry Dakota. Because actually, I think I would probably be down on this end myself in reality. Yeah, let's see. Yes, that's I would be nerd. All right, Dakota, it's us against the world. This is continuous variation. Continuous variation occurs when you have individual genes, each of which contributes a little bit to that phenotype. And so your height is governed by lots of different genes scattered throughout your genome, and each one of those genes contributes a small increment to making you taller. So you have a lot of dominant alleles, which makes you tall. Dakota has fewer dominant alleles, which is why he's shorter. Now there's also environmental effects. Maybe Dakota's parents didn't feed him as much as they fed Brandon. So you can make a tall person shorter by depriving them of nutrients and things like this. But if you have been cared for properly and you reach your genetic potential, then you get the full expression of all of those dominant alleles. And so if you have traits that are determined by lots of different alleles, each one contributing a little bit to your phenotype, you get this type of continuous variation. Not tall and short, but this gradation of continuous variation. Sit back down.
Dakota, did your parents feed you?
Yeah, I was about to say, they deprive you of hair.
So now let's imagine that height is determined by only 3 genes. And each dominant allele contributes 1 unit of height to you. Such that if you are this individual, if you have this phenotype, everybody, let's say that everybody, even the shortest person, in this case, Dakota would be this genotype. He exists, so he can't be a height of 0, so we'll give him a base height of 1. And then for every allele that you have, you get another increment of height. So you would take the base height of 1, add it to these 6 units of height. This individual would be a height of 7. If you have 5 dominant alleles, you would have a height of 6. If you have 4 dominant alleles, you would have a height of 5, etc., etc., etc. So now we don't have to worry about genotypes at all. What do we have to worry about? Number of dominant alleles. Darren, do you have a question?
No, I'm kind of stretching.
Okay. So we can do this, and once again, you'll see symmetry here. 6 dominant alleles, 5, 5, 4, 5, 4, 4, 3. Guess what? 6, 5, 5, 4, 5, 4, 4, 3. 5, 4, 4, 3, 4, 3, 3, 2. 5, 4, 4, 3, 4, 3, 3, 2. Symmetry in this thing. But the symmetry is such that how many individuals have 6 dominant alleles? 1. So if we make a distribution and we plot number of dominant alleles, 6, there's 1 individual. How many individuals have 5 dominant alleles? 1, 2, 3, 4, 5, 6. 5 dominant alleles is 1, 2, 3, 4, 5, 6. 4 dominant alleles. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Have 4 dominant alleles, 3 dominant alleles. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, I've missed somebody somewhere. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. It's supposed to be 20. 15, 16, 17, 18, 19, 20. If you know half of this distribution, what do you know about the other half of the distribution?
You can make it just the same.
It's just the same because the table is the Punnett square is symmetrical. So 2 dominant alleles would be 15. 1 dominant allele would be 5. And 0 dominant alleles would be 1. If everybody starts off with a base height of 1, this would be a height of 1, 2, 3, 4, 5, 6, 7 units tall. What does this start to look like? Hmm? It looks like a bell curve, doesn't it? It looks like a normal distribution. When we do this, what we find is that simple Punnett square Mendelian style genetics underlies these complicated traits that when we look at populations, we see populations that are normally distributed. Whenever you see a normal distribution of phenotypes, you should think, that's a phenotype that is underlain by lots of genes, each dominant allele contributing a little bit to that overall phenotype. Human height is this way, human eye color is this way, human skin color is this way. All of these things have normal distributions when you quantify them, and they're all these polygenic traits, these traits that are determined by multiple genes, each dominant allele contributing a little bit to the phenotype. Questions? Pick up here on Monday. I'm never going to ask you to do a tri-hypercross. As homework, in class, on a test, tri-hypercrosses are just the means of bridging from simple crosses to these kinds of quantitative traits. Because these kinds of quantitative traits are things like the oil content in corn and the amount of milk that a cow produces for these kinds of traits.
Have a good weekend.
Yep, me too.
Keep my food here on.
I will. All right. And I can turn you off.